Hello, class. Last time you studied mitosis. There is another types of cell division which is called meiosis. So today you will compare what is the difference between mitosis and meiosis in terms of chromosome behavior in each phase and different chromosome number after each cell division, also different daughter cell number after each cell division. Heredity is the transmission of a trait from parents to offspring. So what you can receive from parents is a DNA. DNA can be transmitted. That means gene, because genes are made up of a segment of DNA. So you receive DNA half from mother, you receive DNA half from father. And when you receive DNA from parents, sexual reproduction are involved. So we will study about sexual reproductive organ first. Sexual organs are called gonad. So male gonad is called testis. And female gonad is called ovary. Genes are passed to the next generation via reproductive cells called gamete. We call sperm and egg gamete. These gamete can be formed from male gonad and female gonad. For example, testes, male gonad testes undergo meiosis, then they produce sperm. Female gonad, ovary, undergo meiosis to produce gamete, which is called egg. So the gamete, such as sperm and egg, are produced by meiosis from gonad. And then meiosis produce haploid gamete by undergoing meiosis. So do you remember last time you studied diploid? But this time you will study haploid. So haploid is the more related business with meiosis. Okay. When you look at the cell, when you look at inside the cell, there are a chromosome exactly identical to each other. We call this a pair of chromosome, we call this homologous chromosome. So in other words, the cells that have two of each type of chromosome, we call them homologous chromosome. According to homologous chromosome, chromosome numbers in any species can be denoted 2n. This is diploid. For example, chromosome number of a Homo sapiens, such as a human, can be denoted 2n equal 46. So human individual cell hold 46 chromosomes. Therefore, the cells that have a homologous chromosome are called the diploid cell, and then it is identified 2n. Then if you divide 2n by 2 equal n, single n, we call this what? Haploid. Half of diploid. So in case of a human, 2n is 46. Therefore, n equal, you divide 46, divided by 2, become 23. A gamete, sperm and egg have how many chromosome number? Haploid. So there are 23 chromosomes in the sperm. There are 23 chromosomes in the egg, in the oocyte. Oocyte and egg are same cell. Except sperm and egg, the rest part of your body has 46 chromosomes in individual cell. So be familiar with this, the meaning of a haploid and then also diploid. So from mitosis, cell continue to maintain diploid, but from meiosis, the chromosome number reduced in half to form haploid. So again, gamete, sperm and egg contain a single set of chromosome. We call that haploid. For humans, haploid number is n equal 23. Each set of 23 consists of 22 autosomes and a single sex chromosome. So let's think about diploid again. Diploid is 22n equal 46 chromosome. This can be write down 44 plus 2 sex chromosome. In case of a female, it is going to be XX. Right? Now, when you think about haploid, you just divide by 223 equal 22 plus X. Okay. So, oocyte or egg cell has 22 plus X. These are the configuration of a chromosome in the gamete, in the egg. So each set of 23 chromosomes consists of 22. We call the inside the chromosome, except sex chromosome, the rest chromosome are called autosome. So 22 autosome and a single sex chromosome. Okay. This is the case of female. What about the male? In case of male, 2n equal 44 plus x, y. So sperm can carry 22 chromosome plus X, or some sperm can carry 22 plus Y, right? In case of male, right? Therefore, the sex chromosome which determine the sex of individual are called X and Y. In case of female, X. In case of male, X or Y. In an egg, unfertilized egg, the sex chromosome is only X. In sperm cell, the sex chromosome may be either X or Y. Okay? So the remaining 22 pairs of chromosome, this one is called what? Autosome. This one is called autosome. 22 pairs of chromosome. So when you look at this configuration, this 44 is what? Autosome. This part is sex chromosome. Okay, this part is sex chromosome. This part is autosome. Let's solve this question. A typical human body cell contains how many sex chromosomes? 1 sex chromosome, 2, 23, 46. The question is asking about a typical human body cell. Human body cell. They are not asking about the reproductive cell. They did not mention reproductive cell in the question. Therefore, in human cell body, you need to think about diploid, right? Human cell body. Therefore, how many sex chromosomes present in the diploid cell? 2n equal 46 equal 44 plus XX, or 2n equal 46 equal 44 plus XY. At the time, how many sex chromosomes? So 2. This is the answer. And if in the sperm, how many sex chromosomes? Then your answer will be 1. All right, I think you're already familiar of this figure because you study karyotype. Karyotype is the arrangement of chromosome in a way that homologous chromosomes are paired by comparing size, shape, and location of centromere. If you look at the left panel, it shows chromosome number and the karyotype of a female diploid cell. If you look at the right panel, it shows chromosome number and karyotype of a male diploid cell. How do you know? Can you see here? XX. Here's XY, right? So this is chromosome 1, 2, 3, 4, up to 23 pairs. 22, 23 pairs. 23 pairs is composed of XX, right? These homologous chromosomes pair up and then group them, and then scientists put the number. So even though you see single number, it has 2 chromosomes. Number 2 has 2 chromosomes. Number 3 has a pair of a homologous chromosome. Okay. Therefore, if we go by up to number 23, so 44 plus XX. So this is female karyotype. And in this case is male karyotype. So 44, you have to multiply this, 44 plus XY. In this case, 44 plus XX. So you can write down chromosome arrangement as 2n equal 46 to both female and male diploid cell, because there are 46 chromosomes in female and male respectively. But if you want to indicate the sex chromosome in diploid cell, female cells can be denoted 2n equal 44 plus XX, whereas male cell is denoted 2n equal 44 plus XY. Haploid cell result from half of a diploid cell. So diploid cell is divided by 2. In other words, 2n divided by 2 equal n. Okay? So 2n divided by 2 equal n. Therefore, n equal 22 plus x. In this case, n equal 22 plus x or 22 plus y. These are called what? Gamete. So gamete indicate egg, oocyte, and sperm. When oocyte and sperm fuse together, it generates new organism. Only one set is present in the gamete because egg has only haploid chromosome, same as a sperm has a haploid chromosome. Thus, the gamete only has a haploid. When an egg and sperm, each containing half of complement of a chromosome, they fuse together, that it produces a single cell. We call this single cell zygote. And the fusion of a gamete, such as egg plus sperm, we call this phenomena fertilization. In case of non-reproductive cell or somatic cell, they have diploid cells, so they have 2n. 2 sets of chromosomes are present in somatic cell of an adult individual, making them diploid because they already have 2 sets. Why? They receive 1 set from mother, the other set from father by way of fertilization. Let's look at life cycle, the human life cycle. If you look at this cyan color, that cyan color presents haploid life cycle. Haploid life cycle. Okay. And then if you look at this apricot color, that indicate diploid life cycle. So when you look at these women and men, they become mature and then they reach puberty, then they have ability to produce oocyte and sperm from ovary and testes, respectively. When gamete is formed from ovary, they undergo meiosis. Same thing. From testes, when sperm is formed from testes, testes undergo meiosis. Then sperm and egg are formed. These sperm and egg, they are haploid. So you can see sperm have and haploid, egg also has haploid gamete. These egg and sperm, they fuse together. We call that fertilization, and then we call the cell, once sperm and egg fuse together, then we call that cell zygote. Since they fuse, this one haploid plus another haploid become diploid. So the zygote is a single cell. Actually, egg is one cell, sperm is one cell. But when sperm and egg are fused, only this head part of sperm enter egg. And then the rest part is just cut it off. And then inside the head part of sperm, there are DNA. Therefore, once the head part of sperm entered egg, still it is one cell, because the entire sperm cell did not enter egg cell. Therefore, since only portion of sperm cell enter join to egg cell, therefore still zygote is single cell, one cell, but the chromosome is diploid. These zygote undergo what? Mitosis. And then become baby, and then once become reach puberty, they continue to undergo mitosis. The significance of mitosis lies in that somatic cells have a genetically identical configuration. Okay? That is the significance of mitosis. And if you look at our body is composed of somatic tissue and germline tissue. Somatic tissue can be made up by mitosis. Fertilization results in the formation of diploid zygote that begin to divide by mitosis. This single diploid cell means what? Zygote. This zygote eventually give rise to all of the cells of your body, and these cells are called somatic cell or non-reproductive cell. The somatic cells keep doing mitosis to form genetically identical diploid daughter cell. This is, I stressed that last time, the significance of mitosis is going to providing genetically identical configuration of each cell in your body. Otherwise, the tumor can be developed. Compared to somatic tissue, germline tissue is reproductive cell. Okay? Somatic tissue is a non-reproductive cell. Germline tissue, the cell that will eventually undergo meiosis to produce gamete. These cells are referred to as germline cell. Okay? Germline cell. Both somatic cells and the gamete-producing germline cells are diploid. The germline cells undergo meiosis, producing haploid gamete. So I said the sperm can be produced from testes, but testis is not single cell. Testis is organ, reproductive organ. Therefore, this reproductive organ has many cells. Okay? Until testis is fully mature, mitosis keep occur. But once this testis fully mature and has ability to produce sperm, then some cell inside the testis undergo meiosis to produce sperm. By the same token, in order to have mature ovary, mitosis occur. But once female reach puberty, some cell from ovary undergo meiosis to produce egg. Okay, so until humans has a fully developed reproductive organ, they undergo mitosis. But once they reach puberty, then they undergo some different kinds of cell division called meiosis in order to produce reproductive cell, in order to produce a gamete. Okay. So one more time. This time, if we look at this blue color, that is haploid life cycle, and the yellow color is diploid life cycle. So when these adults fully mature and then they produce egg from female, sperm from male, at the time they undergo meiosis. Okay? Then once this gamete, egg and sperm, we call the gamete. Gamete formed, they can fuse, they form zygote. Once egg and sperm fuse, they form diploid cell, which we call zygote. This zygote continue to do mitosis, so it become embryo, fetus, baby, and then keep growing up, and then become adult. Okay? So this life cycle, so your life will undergo mitosis and meiosis, both cell division. But you have to think about that, okay, once zygote is formed, individual cell hold 2N, 46 chromosome. But once they undergo meiosis, chromosome number reduce in half. Okay, think about that, how they can happen. So alteration between diploid and haploid. The pattern of alteration between diploid chromosomes and haploid chromosomes. So from ovary, some female germ-line cells, they hold diploid chromosome 2N, 46 chromosome. Now they undergo, here is the meiosis. Then gamete has 23 chromosome. Okay. It's the same thing to male germ-line cells. Once this egg and sperm fuse, we call that cell zygote, and it has 2N, 46 chromosome. This zygote grown into adult. Why chromosome number need to reduce in half? Think about that. After meiosis, if chromosome number is not reduced, maybe maintain 2N. The same thing in male germ-line, 2N. Then your zygote will have how many? 4N. So in other words, 92 chromosomes inside the cell. Okay, when this adult grown up, and the next generation, so next generation, this person has 4N, and then the other partner also has 4N. If there's no reduction in chromosome number, then the gamete will have also 4N, and here's 4N. Then once these gametes fuse, and then they will form 8N. So inside the cell, chromosome number is about 184. And then this is gonna continue generation by generation by generation by generation by. So inside of the cell, chromosome number is gonna increase every generation. It does not make sense. right? So significance of meiosis is preventing the new organism from having twice as many chromosomes as its parents. Once chromosome number reduced in half, the next generation or the following generation, all humans continually have the 46 chromosome in every cell, but only for reproduction, the chromosome number reduced in half because you need to receive half of chromosome from mother and the rest of half chromosome from father. Let's solve this question. Meiotic cell division in animals occur in where and result in the production of body cell, adult cell, body cell, parent cell, testes and ovary, and gamete, testes and ovary, and diploid cell, which could be the answer. Mitotic cell division in animal occurs in testes and ovary and result in the production of gamete. So number C is the answer. So you're familiar with the diploid and haploid cell. One more time, for humans, the diploid cell, we write down 2n equal 46. If a tiger has 38 chromosomes in the cell, right, then you can write down 2n equal 38. The 46 chromosome, or diploid, 2n, in a human somatic cell are two sets of 23. Each pair of homologous chromosome include one chromosome coming from mother, the other element of pair of homologous chromosome coming from the father. At sexual maturity, the ovaries and testes produce haploid gamete. The gamete are the only types of human cell produced by meiosis rather than mitosis. Only diploid cell can undergo meiosis. Haploid cell cannot undergo meiosis. Haploid cell is the end of cell division. Haploid cell is for fertilization, not doing another cell division. That's why only diploid cell can undergo meiosis. Haploid cell cannot undergo meiosis. Meiosis results in one set of chromosome in each gamete. Therefore, they have only n, 23 chromosomes. Fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. That's why chromosome number reduced in half from meiosis. Let's study a little bit review. Pair of homologous duplicate chromosome. So meiosis operate a little bit different from mitosis, but they also share some commonality in some aspect. So for meiosis, chromosome also need to duplicate. So these are what? One chromosome, and then this is another chromosome, okay? But they are homologous chromosome. Therefore, pair of homologous duplicate chromosome means here, when you look at this chromosome, you can see sister chromatid, okay? And sister chromatid. So each chromosome is already duplicated. And then when they bind these, group them together, that means pair of homologous chromosome. So one more time, if we look at, can I ask how many chromosomes does this cell have? How many chromosomes does this cell have? Can you write down that chromosome number scientific notification, 2n equal what? How many? 2n equal 6. This is correct answer. If you write down 2n equal 12, that is not correct. This is duplicated chromosome, right? But here, this is a centromere. On the centromere, two sister chromatids attach each other, tightly bind. And then I told you that whenever you count chromosome, count the number of centromere. 1, 2, 3, 4, 5, 6. So when you look at this chromosome, you can pull out information such as, okay, here's this, maybe I call this A chromosome has the other partner of a homologous chromosome here. Okay? And then this B chromosome has partner of a pair of homologous chromosome here. That's why they say the pair of homologous chromosome. Okay? And these C, if I see like this, they are homologous chromosome, pair of homologous chromosome. But each chromosome is duplicated. A cell also shows not only homologous chromosome, but also when you look at this duplicated form, these are what? Sister chromatid. This is sister chromatid, correct? But this is a sister chromatid. But when you look at these and these, they are non-sister chromatid. Okay? That's why they say the non-sister chromatid. These and these are sister chromatid. And these are sister chromatid. But between this and this is non-sister chromatid. One more time. Here we go. This is a sister chromatid. This is a sister chromatid. Also, this is sister chromatid. Sister chromatid. But these and these are non-sister chromatid. How non-sister chromatid can come out? Non-sister chromatid can be described between a pair of a homologous chromosome. A pair of a homologous chromosome. Once single chromosome duplicate, so they form two sister chromatid, we just simply call sister chromatid. But when you put these chromosome, duplicated chromosome, and then duplicated chromosome side by side, this and this is going to be what? Non-sister chromatid. Okay, so try to be familiarized with these kinds of naming. Sister chromatid, non-sister chromatid, DNA duplication, chromosome duplication, the same meaning, and homologous pair. And when you look at this red, one set of chromosome which shows red color coming from mother. The other set of blue color chromosome, they are coming from father. Again, diploid cell undergo meiosis to form haploid cell. And this is mother side, and this can be father side. Okay? And then egg and sperm fuse together. We call that fertilization to form zygote. Diploid fertilization, fertilized egg is called zygote. Now we are going to examine meiosis in detail. Similar to mitosis, meiosis is preceded preceded by the replication of a chromosome. You saw that just before. But there's a difference between mitosis and meiosis. Meiosis takes place in two sets of cell division called meiosis 1 and meiosis 2. What does it mean? So once meiosis 1 occurs, immediately another division we call meiosis 2 followed. The two cell division result in 4 daughter cells rather than 2 daughter cells. From mitosis, you only have 2 daughter cells, but from meiosis, you will have 4 daughter cells. From meiosis, each daughter cell has only haploid. So you have to keep in mind, unlike mitosis, meiosis occurs in two sets of cell division, meiosis 1 and meiosis 2. And the daughter cells from meiosis are haploid, and the number of daughter cells are 4, not 2. Okay. So I said meiosis 1 and meiosis 2. And then meiosis 1 is composed of prophase 1, metaphase 1, anaphase 1, and telophase 1. And during meiosis 1, chromosome number reduced in half. So we call this reductional division because chromosome number reduced in half. Meiosis 2 is similar to mitosis because sister chromatid separation occurs during meiosis 2. The result is 4 haploid daughter cell, and then each daughter cell is unduplicated chromosome or unreplicated chromosome. We will get in there more detail. So think about this. A parent cell has 16 chromosomes. How many sister chromatids will be present after duplication of the chromosome? This question is different from question asking if a parent cell has 16 chromosomes, how many chromosomes will be present after duplication of the chromosome? Okay. They are asking the number of sister chromatid. They are not asking about the number of chromosomes after duplication, right? So the answer will be each chromosome produces 2 sister chromatid. Each chromosome will form 2 sister chromatid after duplication. Therefore, the answer is 36. If the question asks about if a parent cell has 16 chromosomes, how many chromosomes will be present after duplication of the chromosome? Then the answer is 16. So do not confuse that. Why? Because the sister chromatid is still tightly attached on centromere, and then I suggest you count the number of centromere as the number of chromosomes. Okay, this is an overview of entire meiosis. So meiosis has meiosis 1 and meiosis 2. Before meiosis takes place, also the cell has interphase. Interphase is composed of G1, S, G2 phase. Right? Do you remember G1, S, G2 phase? And especially in S phase, chromosome duplication takes place. Right? From S phase, 2 sister chromatids are formed. Now this, so if I ask about, look at this cell, how many chromosomes are there? How many chromosomes are there here? If I ask you write down chromosome number with diploid notification, 2n equal how many chromosomes? 2. Okay. And then in this cell, if you write down 2n equal 4, this is correct or wrong? This is wrong. Still 2n equal 2. Why? Here's centromere just bind to sister chromatid. Now, so there are two chromosomes inside the cell. From the meiosis 1, this chromosome number reduced in half. So how many chromosomes you count here? This is one chromosome. Therefore, we cannot say 2n equal 1. This is not correct, because when 2n equal 2, you cannot write down 2n equal 1. This is wrong. So once chromosome number reduced in half, you have to divide the same number to both sides, left and right side. So when you divide 2 by 2 become 1, we have to divide 2n divided by 2 also. So it becomes n equal 1. So it become already haploid. This is what 2n is diploid. So meiosis start from diploid, but in the middle of meiosis, chromosome number reduce in half. And then meiosis 2, you can see that meiosis 1, even though chromosome number reduced in half, still sister chromatid bind each other, right? These sister chromatid separate during meiosis 2. It's similar to mitosis, right? But how many chromosomes exist inside the cell? Still one chromosome. So this n equal 1. Still continue to carry haploid until the end of meiosis. Only difference between meiosis 1 and meiosis 2 is different number of daughter cells. At the end of meiosis 1, there are 2 daughter cells. At the end of meiosis 2, there are 1, 2, 3, 4 daughter cells are formed. Now, one more time, how many chromosomes inside the cell? Just count this centromere, then 2n equal 2, right? After meiosis 1, chromosome become 1, and we call this haploid cell. And then after meiosis 2, still 1 chromosome, so still haploid, but there are 4 daughter cells. Okay. When you look at this one, this is a sister chromatid. Sister chromatid means the chromosome duplicate, right? And then when they bind these together, when they group them together, because they are homologous chromosome. Now, we will study meiosis 1 and meiosis 2 in detail. So meiosis 1 is preceded by interphase. So you saw that the chromosome duplicate to form sister chromatid. Okay. The sister chromatids are genetically identical and joined at the centromere. The single centrosome replicate. Centrosome and centromere, they are different, right? Centrosome, it has 2 centrioles. It has 2 centrioles. And these 2 centrioles formed, and then they migrate to each pole. And then from centrosome, from centriole or centrosome, there's a spindle fiber, some kinds of fiber or string, spindle microtubule produced, and then this spindle microtubule is going to attach it to centromere later. Okay. And then meiosis 1 can be divided into 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, followed by cytokinesis. If you look at prophase 1, this is prophase 1. You can see this homologous pair, and then each chromosome duplicate to form sister chromatid. Okay. So when you look at duplicated chromosome, is pretty much condensed. And then homologous chromosomes pair up. They come together in pair. They are not just freely separate. Look at here, homologous chromosome, they pair up. Here's pair up. Here's pair up, right? These homologous chromosomes come together in pair. And then when you look at these paired homologous chromosome, how many sister chromatids you can count? 1, 2, 3, 4. So this new structure, in other words, the pairing up of a homologous chromosome, consists 4 chromatids. We call this tetrad. So look at here, 2 sister chromatids, 2 sister chromatids. How many chromatids you can count? 1, 2, 3, 4. 4 chromatids is called tetrad. Tetrad. One more time. 1, 2, 3, 4 chromatid we called tetrad. 4 chromatid. And then when you look at these chromosomes, this pair of homologous chromosomes, there's some kinds of crossing over or exchanging some portion of chromosome between a pair of homologous chromosomes. So once this part exchange, then this red color chromosome will carry blue segment of chromosome. And then blue chromosome will carry some portion of red color chromosome. Like this way, right? So we call this chiasmata or chiasma. Chiasma, this is plural, and then singular is chiasma. This is a singular, this is plural. Now you see these, we call this phenomena, we say crossing over red and blue. A pair of homologous chromosomes exchanges some portion of a chromosome. We call this crossing over, and then this site we call chiasma. Now, there's 2 places genetic exchange occur. That kinds of chromosomal behavior can be observed under prophase 1 of meiosis. Prophase 1 of meiosis. So chiasma occur in prophase 1 of meiosis 1. Okay? And the rest thing is nuclear envelope also dismantled. And then centrosome, during interphase, centrosome already duplicated and then start to migrate to each pore. One centrosome move to one pore, the other centrosome move to the other pore. That is the phenomena of during prophase 1. And then when you move to metaphase 1, these, a pair of homologous chromosomes lined on equatorial plane at the center of the plane. And then at the time, the spindle microtubule produced from centrosome is already attached to centromere by way of kinetochore. Yes, spindle microtubule attached to kinetochore. Kinetochore is a protein complex associated with centromere region. So you can see spindle fiber attached to kinetochore in centromere region. Okay, so chromosome line up by homologous pair. That is the chromosomal behavior during metaphase 1. Anaphase 1 is this homologous chromosome separate. One member of each pair of homologous chromosome go to one pore, and the other member of a pair of homologous chromosome move to the other pole. Spindle microtubule drag each member of homologous chromosome to both poles. Therefore, homologous chromosome now separate. Each pair of homologous chromosome become separate. Okay, that occurs during anaphase 1. Therefore, if you see telophase 1, you can see two nucleus present inside the one cell because the cytokinesis has not occurred yet. But if we look at inside the two nuclei condition, inside each nuclei has already half chromosome number, haploid. And then two clusters of chromosome have formed, and then each cluster contains one member of each pair of homologous chromosome. Therefore, the daughter nuclei, this is already haploid. Now, the spindle microtubule is disappear. And then look like two nuclei inside one cell. Then immediately cytokinesis occur. Do you remember cytokinesis? Microfilament protein cytoskeleton is going to constrict the microfilament ring toward the center. Therefore, eventually these two, the cytoplasm is going to divide into half. Therefore, you will have two separate cells like this. So you finish meiosis 1. And from meiosis 1 to this is meiosis 2. Between these, there's no interface. After meiosis 1, meiosis 2 occur immediately. So there's no interface. There's no interface means there's no chromosome duplication. Since there's no interface between meiosis 1 and meiosis 2 immediately take place right after cytokinesis of meiosis 1. Meiosis 2 also consists of prophase 2, metaphase 2, anaphase 2, and telophase 2 followed by cytokinesis, another round of cytokinesis. Prophase 2, the spindle microtubule form again. Do you remember? In telophase 1, spindle fiber disappear, but in prophase 2, this spindle fiber come back again. So spindle microtubules reform and attach to the sister chromatid, this time sister chromatid. Homologous pairs separate during meiosis 1. So you cannot see a pair of homologous in the daughter cell during meiosis 2. They already separate. But you can see sister chromatid. And on this sister chromatid, microtubule, spindle microtubule try to attach it to the sister chromatid. And metaphase 2, the chromosome line up along the equator. The sister chromatids of each chromosome are attached to spindle microtubule by a kinetic core, and the microtubule leads to opposite pole. Okay, this microtubule leads to opposite pole. Anaphase 2, the chromatids separate into independent daughter chromosome. So see, this chromatid separate, and then one chromatid move to one end, the other chromatid move to the other end. Chromosome themselves cannot move, but actually spindle microtubule pull chromatid toward their pole. Telophase 2, the chromosome finish moving to opposite pole. And then nuclear envelope reformed, and then chromosome decondense. So it becomes chromatin. And then immediately cytokinesis occur. Therefore, one 2, 3, 4 daughter cells are formed. And each daughter cell contains one member of each pair of homologous chromosome. This is the end of meiosis. So one more time, prophase 1 of meiosis 1. Prophase 1 of meiosis occupies more than 90% of the time required for meiosis. Chromosome become condensed. In synapse means a pair of homologous chromosomes come in together. And then they pair up. They align side by side. We call that synapse. Each pair of chromosomes form a tetrad because one chromosome has 2 sister chromatids. And a pair of chromosomes means 2 chromosomes. The other member of a pair of homologous chromosomes also has 2 sister chromatids. So you have all 4 sister chromatids. We call this what? Tetrad, a group of 4 chromatids. In crossing over between non-sister chromatid of a pair of homologous chromosomes, crossing over takes place. Non-sister chromatid exchange DNA segment. So crossing over occurs between non-sister chromatid of a pair of homologous chromosomes. Each tetrad usually has 1 or more chiasma or chiasmata, the same meaning. Chiasma, chiasmata, it appears as X-shape because 2 non-sister chromatids cross each other. We call it crossing over, so it looks like an X-shaped region where crossing over occurs. So we will see. Here's a pair of homologous chromosomes, right? And then each chromosome is duplicated. So here's sister chromatid, and then here's sister chromatid. And then crossing over never ever occur between sister chromatid. Crossing over never ever occur between sister chromatid. Crossing over occur between non-sister chromatid. So if we call these and these sister chromatid, these and these are what? Non-sister chromatid. Non-sister chromatid. Okay? Between non-sister chromatid, there's crossing over occur. The site of crossing over is called chiasma or chiasmata. Chiasma, same meaning. See, can you see this X portion? Okay, here's X portion. So once crossing over occur, once chiasma formed, then part of chromosome exchange between homologous chromosome. Can you see? The beginning, it was entire yellow color. Now it has portion of purple DNA segment. When you look at purple chromosome, now you can see portion of yellow chromosome portion, and then you can see yellow chromosome segment. So some chromosome portion exchange, swapped. We call that chiasma at the site of crossing over. So crossing over is kind of a phenomenon. And then we call that site chiasmata or chiasma. One more time. This is sister chromatid. This sister chromatid. And this non-sister chromatid, even though they are a pair of homologous chromosomes. Okay. Now, between non-sister chromatid, crossing over occur. Can you see X shape here and then X shape here? Synapse occur. Synapse means associate and attach it. Now, once this crossing over occur, some chromosome segment can be exchanged. So when you look at, see, this chromosome can carry coming from the other partner of homologous chromosome. So this crossover occurs between non-sistochromatid of a pair of homologous chromosomes, must occur within a homologous chromosome. But even though it occurs between homologous chromosome, it should be non-sistochromatid, not within sistochromatid. So one more time, in synapse, there is one more word, synapse. Homologous chromosome loosely pair up, aligned gene by gene. So this gene, there's some gene in this location, and in this location is exchange here. There's some gene located here, there's some gene located here, and then those genes can be exchanged. Each pair of chromosome form tetrad. We call this tetrad. Because there are 4 chromatid. A group of 4 chromatid, and then non-sistochromatid exchange DNA segment here in a process called crossing over. Each tetrad usually has 1 or more chiasmata. So here's 1, 2. Okay, sometimes more than 2, sometimes only 1, depending on, a chromosome also responds to environment. So we have no way how many chiasmata will form between non-sistochromatid. X-shaped region where crossing over, this is the crossing over, the site for crossing over occur, and then we call this chiasmata or chiasma. So this is the information written in expression. So in metaphase 1, tetrad, 4 sistochromatid line up at metaphase plate, equatorial plate, at the center of the cell, with 1 chromosome facing each pore. Spindle microtubules from 1 pore are attached to the kinetochore of 1 chromosome of each tetrad. Microtubules from the other pore also attached to kinetochore of the other chromosome. Anaphase 1, pairs of homologous chromosome separate. See, the separation of a homologous chromosome. That is main event in anaphase 1. 1 chromosome moves toward each pore, guided by the spindle apparatus. Sistochromatids remain attached at the centromere and move as 1 unit toward the pore. Now, from this slide, anaphase 1. I hope students just erase for this one because there's no any contents about metaphase 1 in this slide. So, anaphase 1, homologous chromosomes separate, but cystochromatids remain attached. A cystochromatid has not separated yet. Only a pair of homologous chromosomes say bye-bye. That is the event in anaphase 1. So look at here, prophase 1, the 4 chromatid, in other words, tetrad, coming together, they pair up. A pair of homologous chromosome pair up. See? And also what happened here? This is crossing over here. So this is the site of crossing over, which is called chiasma. Chiasma. Only one place we call the chiasma. If we see more than one, we can say chiasmata. And then can you see here, chiasma? 1, here's chiasma. But look at here, chiasma only occur between non-cystochromatid of a pair of homologous chromosomes. Chiasma cannot occur from between different homologous pair. Now, prophase 1, each homologous pair undergo synapsis. Then synapsis means they attach tightly, they pair up. And crossing over between non-cystochromatid with the subsequent appearance of chiasmata. Now, metaphase 1, chromosome line up as a homologous pair on the metaphase plate, equatorial plate. Anaphase 1, homologous chromosomes separate from each other. But when you, so these homologous pair, they separate. But when you look at each chromosome, two sister chromatids still remain joined at the centromere. Can you see why each chromosome has some mosaic color? Because the crossing over occurred during prophase 1. That crossing over or chiasma continue to metaphase and anaphase. Now one more time. This is a pair of a homologous chromosome. And then you can hear this is a sister chromatid. And then between these and these chromosomes, we call non-sister chromatid. And if you look at this part, this is the location of gene 1. This is the location of gene 2. You need to have a gene for your eye color. You need to have a gene for hair. You need to gene for earlobe. So many physical appearance depend on the gene. So this is the location of maybe eye color. This is the location of gene for hair. And then the other element of a pair of a homologous chromosome also has same location for gene 1 here and gene 2. We call that allele. This is a pair of a homologous chromosome and then same location, but the genetic content can be a little bit modified. We call that allele. This allele will come back later when you study genetics. But I just want you to know when you have some concept, this allele can be exchanged. Okay? So, for example, if I say gene 1 is for, this gene is for eye color, then this chromosome has eye color for blue eye color. This chromosome carry brown eye color. Once they form crossover here, originally yellow chromosome has blue eye color and purple chromosome has brown eye color. But from this crossing over, they can change. This yellow chromosome can carry brown eye color. Also, the purple chromosome can carry blue eye color. Because of a crossing over chiasmata, some gene can change their location. That is important concept when you study allele down the road. All right, let's go back to meiosis. Meiosis 1 is a telophase. We complete anaphase 1, the homologous chromosome separate. Okay? And in the beginning of telophase 1, each half of the cell has a haploid cell because the chromosome number reduced in half already occur in anaphase 1. So from telophase, then each chromosome still consists of 2 sister chromatids. And from telophase, there are 2 daughter cells formed after meiosis 1, right after complete cytokinesis. So cytokinesis occurs simultaneously forming 2 haploid daughter cells. In animal cell, cleavage furrow forms. In case of a plant cell, cell plate form. So forming their cell plate is different in case of animal from outward inward, and in case of plant is from inward toward outward. So no replication, chromosome replication occur between the end of meiosis 1 and the beginning of meiosis 2 because chromosomes are already duplicated. 2 sister chromatids remains after meiosis 1. This means still duplicated condition. So let's look at meiosis 2, also composed of prophase 1, 2, metaphase 2, anaphase 2, telophase 2, followed by cytokinesis. Meiosis 2 is pretty much similar to mitosis. What is the main characteristic of mitosis? 2 sister chromatids separate. That event occurs during meiosis 2. In meiosis, each daughter cell is genetically distinct from the others and from the parents. We will see about that. Mitosis. Each daughter cell is genetically identical from parent cell. But in meiosis, each daughter cell cannot be identical from parent cell because this chiasma crossing over occur during prophase 1 of meiosis. So the chromosome already exchanged some portion with the other pair of homologous chromosome. Therefore, after meiosis, each daughter cell has a distinct genetic configuration according to chiasmata crossing over. We will see that. Prophase 2, the spindle fiber, spindle microtubule reform and attach to sister chromatid, metaphase 2, they align at equatorial plate, and then spindle microtubule is going to drag chromatid to both ends, and then anaphase 2, or sister chromatid separate. And telophase 2, there are nuclear envelope reform, and then spindle microtubule disappear, and then but each daughter cell has only one member of each pair of homologous chromosome. Okay. So during another round of cell division, the sister chromatids finally separate for haploid daughter cell. Each daughter cell containing unduplicated chromosome. Okay, so you can see 1, 2, 3, 4 daughter cell, and then each daughter cell has how many chromosome? Haploid. So let's solve this question. Which of the following events does not occur in prophase 2, but does occur in only prophase 1? Prophase 1, you have a pair of homologous chromosome. Therefore, synapses occur, yes. Also, crossing over occur. I think the close answer is more like crossing over and synapses only. I think prophase 2 also spindle formation take place. Yes, but they are asking about only the event can occur in prophase 1. Prophase 2, this crossing over and what? Crossing over and synapses cannot take place. So even though this spindle formation might be correct, but this and this cannot occur. Therefore, the D cannot be the answer. But this is enough, sufficient answer from this question. Only synapse is correct, but crossing over also correct. So include synapses and crossing over together. This is the best answer. This is the best answer. And then prophase 2, there's no crossing over, there's no synapses formed. Therefore, even though there's a spindle formation appear on D, this cannot be the answer. So the answer is E. So this is another description about each phase of meiosis 2. So you can read through what is the main event in prophase 2, metaphase 2, and anaphase 2. It's pretty much similar to mitosis in terms of separation of sister chromatid. Okay. Now, telophase 2, and then nucleoform and chromosome begin decondensing to form chromatin. Okay. And telophase 2, immediately cytokinesis occur. Therefore, there are how many daughter cells from meiosis? 4 daughter cells. And each daughter cell has haploid set of unduplicated chromosome because the sister chromatid already separated during anaphase 2 of meiosis 2. So it becomes unduplicated chromosome in telophase 2. And then each daughter cell is very distinct from one another and even distinct from parent cell. So look at this during metaphase 1. Look at here. If I ask about the chromosome number equal how many? 1, 2, 3, 4, 5, 6. 6 chromosome. And then you can see 3 set of homologous pair, right? This and this homologous, another set of homologous, another set of homologous chromosome. Now, when a pair of homologous chromosome can align at the center of the cell, which set of homologous chromosome has what position? This is a possible chromosome arrangement at metaphase of meiosis 1. Maybe they can have this position, or this purple and yellow, they can swap the position. Well, this time, the second set of homologous chromosome, they can change the position. They might have this arrangement. So there's no way we cannot put our hands into the cell and then reorganize the position. That's impossible. Therefore, we do not know which member of a homologous chromosome pair faces toward which pole. So this is one pole. This at the if you think this as a cell, a set of homologous chromosome is going to move to one pole. The other homologous chromosome partner is going to move to the other pole. Then after meiosis 1, each daughter cell has this set of homologous chromosome. Every cell has different combination of a set of homologous chromosome. So 8 possible sets of homologous after meiosis 1. Now, if we look at panel C, after meiosis 1, meiosis 2 occur, and then these continue to give 8 possible type of gamete after meiosis 2. So these gamete might use for fertilization. Every gamete has potential to form zygote. Therefore, you never know which gamete you are going to be the partner of the other gamete of the other partner. So this is showing genetic variability. Even though you share the same parents, your siblings has a different, in some sense, you look after each other, but you have some unique physical appearance. Because in case of 6 chromosomes, there's 8 different possibilities to become gamete. You might choose this gamete to form zygote. Maybe your sister might choose this gamete to form zygote. Maybe your brother might choose this gamete to form zygote. That's why in this world, everyone has uniqueness except identical twin. So you have 8 kinds of possible type of gamete. So how you can calculate possible combination? If you have 6 chromosomes, n equal 3, and then you will put these 3 as exponent. Okay, then this is going to be 2 times 2 times 2 times equal 8 possible types of gamete. Okay. So this kind of random orientation of a chromosome is called independent assortment. Independent assortment. They have freedom. They are independently assort each other. So kinds of shuffling the homologous chromosome create novel combination of chromosome. Just change the chromosome position. These kinds of shuffling homologous chromosome create a novel new combination of chromosome. Right. So mitosis conserves the number of chromosome set. For example, diploid to diploid, producing cells are genetically identical to the parent cell. Meiosis reduces the number of chromosome set from diploid to haploid, producing cells that differ genetically from each other and from the parent cell. Okay, one more time. Now we are compared between mitosis and meiosis. Okay, the parent cell. So how many chromosomes? 1, 2, 3, 4, 5, 6. 2 n equals 6. Right? And then even before duplication occur, you can find homologous chromosome. This and then this homologous cell. And this homologous chromosome. This and this, a pair of homologous chromosome, right? From mitosis, prophase 1, the chromosome duplicate. Meiosis 1, prophase 1 of meiosis, also chromosome already duplicated. This duplication occur before these cell division begin. Before M phase, there is inner phase. Do you remember? Inner phase. During S phase, chromosome duplicate. So from S phase, they already formed sister chromatid. Now, when you look at prophase, okay, meiosis 1, during prophase 1, a pair of homologous chromosomes pair up. They attach side by side, right? At that time, crossing over, a chiasmata occur by way of crossing over. Right? And then mitosis, metaphase, they just line up at the equatorial plate. And the same thing, metaphase 1, a pair of homologous chromosomes line up at the center of the cell. Now, mitosis, anaphase, and telophase, the sister chromatid just separate and then move into daughter cell, two daughter cell. And each daughter cell has same chromosome number as parent cell has. But meiosis 1, a pair of homologous chromosome pair up. Now, not pair up, aligned at the center of the cell. Now this, a pair of homologous chromosome separate. So anaphase 1, in telophase 1, the chromosome number becomes reduced in half. Can you see this haploid n equal 3? And this, after telophase 1, of course, there is a cytokinesis, and then immediately meiosis 2 begin. And meiosis 2 is similar to this mitosis. So at the end of meiosis 2, there are 4 daughter cells produced, and each daughter cell has only one set of homologous chromosomes. So you can read this, and then DNA replication occurs during inner phase, okay? Also same thing, inner phase. Now number of division, just one time. Meiosis 1 and 2, therefore 2 times. Synapses, no occur. But synapses occur during prophase 1. And at that time, crossing over occur between non-sister chromatid, resulting in chiasmata, okay? And then still sister chromatid attach each other. The number of daughter cells, 2 daughter cells produced from mitosis, 4 daughter cells produce meiosis, and then each daughter cell has haploid. In case of mitosis, there are two daughter cells, but each daughter cell has diploid. And from mitosis, each daughter cell has genetically identical. In case of meiosis, each daughter cell has different genetic configuration compared to parent cell and even compared to daughter cell each other. Role in the animal cell enable multicellular adult to arise from zygote. Growth, repair, development, okay? And in case of meiosis, produce gamete, reduce number of chromosomes in half, introduce genetic variability among the gamete. One more time, look at the chromosome number. Meiosis starts from diploid 2n, but after meiosis 2, actually after meiosis 1, they have the chromosome number decrease in half. And mitosis is maintained same chromosome number. And it actually, this is for making oocyte egg. This is making sperm. And then there is a several round of cell division eventually produce oocyte and sperm. But in case of a female, there is a tissue named oogonium. This oogonium undergo mitosis, and then they form primary oocyte. And then this primary oocyte undergo meiosis. And then after meiosis, actually there are 4 daughter cells, but 3 daughter cells after meiosis 2 is going to be degenerate. Okay? So it's not active. Only one single oocyte can function. In case of sperm, spermatogonium is the diploid cell, and then it starts to undergo meiosis to form primary spermatocyte, and then they undergo meiosis, and then they have one spermatogonium produce 4 sperm cells. Okay, so one spermatogenesis, each sperm can be functional. But in case of a female, there are 4 daughter cells formed after meiosis 2, but 3 daughter cells become degenerate, so only one oocyte can be functional. So there is some uniqueness to meiosis compared to mitosis. That is synapses and crossing over in prophase 1. At a metaphase plate, in other words, equatorial plate, there are paired homologous chromosomes instead of individual replicated chromosome. Another uniqueness is homologous chromosomes physically connect and exchange genetic information by way of crossing over to form chiasmata or chiasma. At anaphase, it is a homologous chromosome instead of a sister chromatid that separate during meiosis. So sister chromatid adhere tightly until throughout meiosis 1. Now I mentioned that genetic variation or genetic diversity produced in sexual life cycles contribute to evolution. Mutations are the original source of genetic diversity. The gene, this nitrogenous base sequence can be changed according to environmental change. When you have too much UV radiation, also DNA base sequence can be changed, or maybe you might lose some DNA base. So mutation can take place. So mutations are the original source of genetic diversity. Mutations create different versions of a gene called allele. Allele, I introduced a little bit before. Allele is like, for example, you have, this is a simile, maybe you have the gene for red hat, but your brother has the gene for blue head. We call that allele. Both has gene for head, but your head is red color. Your brother's head shows blue color, but both are stand for the gene for head. We call that allele. Allele is a little bit isoform of the gene. Okay? Different version of the same gene. So reshuffling of allele during sexual reproduction produce genetic variation. The behavior of chromosome during meiosis and fertilization is responsible for most of the variation that arise in each generation. So 3 mechanisms contribute to produce genetic variability. One is independent assortment of chromosome. Do you remember? A pair of homologous chromosomes, their position can be shuffling. Okay? Therefore, each daughter cell has a different combination of a set of homologous chromosomes. And also, inside a pair of homologous chromosomes, crossing over occur. There's no fixed rule to generate crossing over. It's a little bit random event. Sometimes between non-sister chromatid of a pair of homologous chromosomes, there might be no crossing over take place. Sometimes more than 3, more than 4 times crossing over can take place. Therefore, the outcome, when you look at the daughter cell from meiosis, each daughter cell has different combination of different DNA configuration of in the one set of a homologous chromosome. Also, which gamete you are going to have to form zygote, that is random fertilization. So these 3 conditions produce genetic diversity in this world. Okay, this is the location of a gene, right? Gene. Once duplicated chromosome, the gene location, there's no change. Now, here's allele, different allele, this part and this part, you can see different allele, even though they can group into same homologous pair. Right? And then this crossing over occur, therefore, the chromosome, the allele position changes. So when you look at the outcome, all these 4 chromosomes, they will enter to daughter cell, and then each daughter cell will have different genetic configuration. Okay. Let's see. Okay. Some location of certain gene, we call that gene loci. Loci means plural, or single is gene locus. Okay? The single one is gene locus. And then this chromosome comes from mother, the father. This chromosome comes from father. Right? They become a pair of homologous chromosome. Okay? Both chromosomes carry the same allele of the gene at this locus. See, single locus. The organism is homozygous at this locus. The same color. Also, look at here. This gene locus contains another gene for which the organism is homologous. But what about, look at here. Each chromosome carries different allele, different color, right, of this gene. So the organism is heterozygous at this locus. In other words, this is might be eye color gene, is red eye color gene. This might be blue eye color gene. So you can have heterozygous gene, and then which is dominant. blue eye color gene is dominant to red eye color gene, then your eye will show blue eye color. So these kinds of genetic variation can occur during prophase 1 when the non-sister chromatid undergo a crossing over or shuffling of a pair of homologous chromosome at metaphase 1. And a pair of homologous chromosome, how these a pair of homologous chromosome determine their position. And all these conditions will produce more and more genetic diversity. So independent assortment chromosome. So homologous pair of chromosome orient randomly at metaphase 1 of meiosis. Independent assortment, each pair of chromosome sort maternal and paternal homologue into daughter cell independently of the other pairs. So let's look at the figure. This one more time. This figure shows kinds of reshuffling of a pair of homologous chromosome at metaphase 1. So as I said, we do not know which member of a homologous chromosome pair face toward which pole. This time in this cell, yellow chromosome goes to same daughter cell with the rest yellow chromosome. In this cell, one purple chromosome goes with two other yellow chromosome into the cell, right? Like this. Therefore, we have no idea which member of a homologous chromosome pair face toward which pole. It's a random orientation. In these kinds of independent assortment, each pair of chromosomes sort maternal and paternal homologue into daughter cell independently of the other pairs. Therefore, the number of combinations possible when the chromosome assort independently into gamete is 2 power to n. So like this. So how we can estimate the possible combination number? Then we can calculate chromosome assort independently into gamete is 2 exponent n. N is a haploid number. In case of a human, n equal 23. So this number, you have to multiply 2 by 2 by 2 by 23 times. It gives 8 million possible combination of chromosome. This number is more than enough to produce genetic variability. So this is a simple example. Possibility 1, there is a 4 chromosome, right? And then after meiosis, you have 4 daughter cells, and each daughter cell has these kinds of chromosome combination. The other possibility has you have different chromosome combination. In this cell, in this daughter cell, you have a long blue chromosome and a short red. Okay? The other combination has a long red chromosome and then a short blue chromosome. So you will have different combination of daughter cell in terms of independent assortment of homologous chromosome. So also, in addition to independent assortment, there's crossing over occur. This crossing over produce recombinant chromosome, which combine DNA inherited from each parent. So in crossing over, homologous portions of 2 non-sister chromatids trade each other, okay? Trade chromosome segment. Therefore, crossing over contribute to genetic variation by combining DNA from 2 parents into a single chromosome. Okay. This crossing over also contribute to generate more genetic diversity. Now one more time, the crossing over occurs always between non-sister chromatid. And then there's chromosome portion exchange. And then if you look at the daughter cell, each daughter cell has all different DNA configuration. So crossing over contributes to genetic variation by combining DNA from two parents into a single chromosome. Also, the random fertilization adds to genetic variability because any sperm can fuse with any oocyte. The fusion of two gametes, each with a 4.8 million possible chromosome combination from independent assortment. Do you remember? This number comes from 2 power 2, 23. Produce a zygote with any of about 70 trillion diploid combination. Okay. This is really enough number, enough probability to make yourself as a unique creature. Crossing over adds even more variation. Each zygote has a unique genetic identity. This genetic variation really connected to evolution. Okay? Natural selection results in the accumulation of genetic variation favored by the environment. Sexual reproduction contributes to the genetic variation in a population which originates from mutations. This content will come back when you study evolution later. Now, you study mitosis and meiosis. But do mitosis and meiosis occur without mistake? How do errors in meiosis cause human genetic disorder? If something wrong during mitosis or meiosis, does that error impact human health or bring some human disease? Do you remember M checkpoint? There are 3 checkpoints when you study mitosis, the cell cycle. M checkpoint means in the middle of cell division, when sometimes spindle microtubule cannot attach to kinetochore properly. So sometimes when spindle microtubule cannot drag the chromosome to one pole properly, then what happened to the chromosome number in daughter cell? Okay, at the time, M checkpoint is going to stop the process of cell division. But sometimes even that M checkpoint can be what? Loosely checked. Therefore, the cell division might just progress with mistake. So, non-disjunction of a sex chromosome in males or females produce abnormal number of X and Y chromosome. Non-disjunction is a new word. Non-disjunction, that means it's not proper segregation. It's not proper separation. Okay? So non-disjunction of a sex chromosome in male produce sperm with no sex chromosome. We indicated this as O, the alphabet O. Okay? Or 2 sex chromosomes. Sperm might have XX, YY, or XY. Okay? Sperm is supposed to have either one X or one Y, but sometimes the sperm can have XX, XY, YY too. So non-disjunction of a sex chromosome in female can produce eggs that are no X chromosome, or XX chromosome instead of one X chromosome. So normal female egg should have one X chromosome, but sometimes some egg has 2 XX, or some egg does not have even single X chromosome. when normal gamete fuse with this defective sperm or eggs, the zygote have a normal number of autosome, but abnormal number of sex chromosome, because there is some problem occur on sex chromosome, not on autosome. The most common abnormalities are XO, XXX, XXY, and XYY. We can put this category as mutation, but this is a severe mutation. Mutation means sometimes the DNA-based sequence can be changed. We call that mutation. That is a little bit a small level of mutation. The changing of chromosome number is higher level of chromosome, and then it can be serious. So normal meiosis. How many chromosomes in this cell? 2n equal 4. 1, 2, 3, 4, all 4. So normal meiosis, non-disjunction during meiosis 1, non-disjunction during meiosis 2. Okay, but the beginning start point is normal. So parent cell has 4 chromosomes or category, all 3 categories. And then meiosis 1, the normal meiosis, in case of that, the meiosis 1, they have daughter cell has haploid chromosome. And then after meiosis, each daughter cell has, haploid, therefore this should be 2. This should be 2, right? So can you see 1, 2, 1, 2, 1, 2, 1, 2. Each daughter cell has 2 chromosomes. So this is just normal meiosis. No mistake, no error. But when you look at non-disjunction during meiosis 1, look at here. Now, a pair of homologous chromosomes supposed to align at equatorial plate, and then a pair of homologous chromosomes supposed to separate to each pole, toward each pole. But in this pair of homologous chromosomes, they, no problem. One member of homologous pair move to this pole, the other member move to the other pole, good. But the other homologous pair, they did not separate. They do not separate, and they move to same pole. So after meiosis 1, one daughter cell has 3 chromosomes. The other daughter cell has only 1 chromosome. And then this condition continue to undergo meiosis 2. Therefore, when you look at daughter cell from this cell, then they will have n equal 3, n equal 1. Okay? And then each daughter cell has potential to form zygote. Okay? Each daughter cell will fuse with gamete from the other partner, right? Then what will happen? The chromosome number of that zygote. One more time, non-disjunction during meiosis 2, the starting point is 2n equal 4, and then meiosis 1 occur normally, no mistake, but during meiosis 2, when sister chromatid separate, this cell is correct, no error. But in this daughter cell, 2 sister chromatid move to same pole. Therefore, after entire meiosis division, then this daughter cell will have 3 chromosomes. This one has 1, and then the other has 2, both. So if this cell participates for fertilization, then the next generation, there will be no problem. But if this gamete or this gamete is going to fuse with other gamete from partner, then the chromosome number of zygote will be changed. So this is non-disjunction means the members of a chromosome pair fail to separate. So the number of chromosome pair cannot separate correctly. We call that non-disjunction. Non-disjunction means the members of a chromosome pair cannot separate correctly. So here, chromosome pair cannot separate. See, therefore, the daughter cell will have abnormal chromosome number. Here, a pair of chromosome or even sister chromatid cannot separate correctly. Therefore, the daughter cell will have abnormal chromosome number. Okay, we call this non-disjunction. So this is an example in case of a human. Once non-disjunction occurs on the sex chromosome during meiosis, what kind of disorder human can have. Okay. We will see figure, and then it might be. Let's solve this question. When a gamete fuse during fertilization, zygote formed. Now you more familiarize this word and the word fertilization. Okay. Now continue to this non-disjunction. How do errors in meiosis cause human genetic disorder? Okay. So the genes on the X chromosome are so essential to survival that any embryo without at least one X chromosome spontaneously abort very early in pregnant. You will see like this. This is diploid. Diploid. So it's going to be 44 plus XX. Okay. In case of a male, it starts from diploid. 2n equal 44 plus XY. Now once this diploid undergo meiosis, then they will have n equal 22 plus X, right? Haploid, n equal 22 plus Y, right? These are the gamete. And this gamete supposed to be n equal 22 plus X, but this time n equal 22 plus XX, right? And then in this case, n equal 22. And then there's no, there's no X chromosome. Okay, we write down then this as 0, not 0, O, like that, right? And then if these are the gamete, right? In other words, egg, this is a sperm. When this egg and then this sperm fuse to form zygote, then you just add up them. 2n equal 44 plus XXX. In this case, if this egg fuse with this sperm, is going to be, just add up these and these, 2n equal 44 plus 1x, coming from this male gamete, okay, sperm. No sex chromosome. Only this X chromosome comes from male gamete, sperm. When this gamete fuse with this male gamete, then you just add up 2n equal 44 plus XXY, XXY. In this case, 2n equal 44 plus YO. So this has how many chromosomes? 45 chromosomes. This has how many chromosomes? 47. Here's 47. We are supposed to have 46 chromosomes. Then 45 chromosomes. It's really hard to survive, except Turner syndrome. Except this case, if the zygote has 45 chromosomes, it's really fatal. But in case of 47 chromosomes, It's more than 46 chromosomes. So you might think that, oh, more might be better. That's not true. They have one more chromosome, but that gives some kind of disorder. We will study that. So non-disjunction can occur meiosis as well as by way of mitosis. So this is mitosis non-disjunction. So far, we saw that the non-disjunction occur on a sex chromosome. Non-disjunction can be resulted from meiosis, but non-disjunction can occur by way of mitosis. So this is 2N, and then this mitosis is supposed to have identical genetic configuration. So, okay, but from this cell, when this cell undergo mitosis, maybe spindle, microtubule drag the two cystochromatid to one pore together. Therefore, the chromosome number, the abnormal chromosome number, can be formed. That in case is 47, and then it has 44 plus XYY. And the other daughter cell will lose one sex chromosome to the other daughter cell. Therefore, 2N equal 45 plus XO. I'm sorry, this is not 45. 44, I'm sorry, 44 plus XO. Therefore, total is 45. Okay? Because 44 are orosome. And then the rest two supposed to be sex chromosome. In this daughter cell, it has only one sex chromosome, not two sex chromosomes. In this cell, it has more than three sex chromosomes. So these kinds of abnormal chromosome number can result from mitotic non-disjunction. At the time, this 44 plus XYY can be formed. So in case of Turner syndrome, female has N equal 22 plus no sex chromosome. And in case of male, has a gamete 22 plus X. Okay. Once they fuse together, they form 2N equal 44 plus XO. So Turner syndrome XO occurs in female with only one X chromosome. See, you have only one X chromosome. So total chromosome number is what? 45. So Turner syndrome means incomplete or missing one X chromosome on 23rd chromosome. At puberty, hormone deficiencies prevent this Turner syndrome from menstruation or developing a secondary sexual characteristics. So it affects women the lack of a mature egg, so they remain infertile. Additional symptoms include a short stature, and the skin around the neck area is fold, and then increased risk of cardiovascular disease, kidney defect, and hearing loss. So another human disorder resulted from non-disjunction is trisomy X or triple X is the same meaning. So this is N equal 22 plus XX. So it has how many chromosomes? Haploid 24. And here's is a normal. N equal 22 plus X. So if you combine them, you will have a 44 plus XXX. These three copies of X chromosome is called trisomy or triple X. It's the same meaning. Is normal woman with an extra X chromosome, and there's no any significant symptom can be shown. And there is an increased chance of learning retardation and then tendency toward tallness associated with trisomy X. In other words, if female with triple X, those females shows really very, very tall. Women with trisomy X bear normal XX and XY children. So in terms of reproduction, there is no much problem for reproduction. So one more time, trisomy, it has 3 copies of X chromosome. Female is very tall, and then the ratio to have trisomy X symptom is 1 out of 1,000. And then Turner syndrome is missing X chromosome. So they cannot reproduce offspring, and then short stature, and then the sex organ is now fully developed. So if we look at this diagram, the left woman shows trisomy X. It's very tall. Okay. Based on external appearance, it's not really clearly distinguished from normal XX female, except by karyotype. But when we take out blood from this triple X woman and then analyze the karyotype, this woman has 1 more X copy, sex chromosome. And then, but the only difference is very, very tall. But even with the normal XX chromosome, 46 chromosome with the 2 XX chromosome female, they can be very tall. So, when we look at by naked eye, we have no way to distinguish normal female woman and then trisomy X woman. And then the right woman shows this is Turner syndrome. Okay? In other words, there's no X chromosome. So 44, only 1 X chromosome. There is 1 missing X chromosome. Okay? So total chromosome number is 45. Okay? A sterile female with a short stature. And then let's look at this Turner syndrome like this. Turner syndrome. Can you see this area? This around the neck area? And then neck area, between neck and shoulder, you can see a little bit, a web of skin extend. A web of skin extend between neck and shoulder. Okay? And then the breast is very poorly developed. And also, Turner syndrome is the only case where having only 45 chromosomes is not fatal in humans. So, in other words, if someone has like 44 plus, this one, it cannot be survived. But Turner syndrome is 20 equal 44 plus X. So still have a 45 chromosome, right? Both cases had 45 chromosome. But in this case, it's not survivable. But only this Turner syndrome can survive with chromosome number 45. With only 45 chromosome, still is not fatal in humans. Monosomic zygote, that means only 1. Mono means 1. Only a monosomic, but only 1 copy of a particular chromosome. Only X chromosomes are there. See here, XO. Okay, I told you that the mitotic non-disjunction, I explained that, and then in this case, we call this Jacob syndrome. Jacob syndrome have extra Y chromosome. Okay, men with Jacob syndrome have high levels of testosterone and maybe exceptionally tall too. Okay? And also it does not really show any well-defined syndrome, okay? Except that they tend to be very tall. So, but this non-disjunction can occur through mitosis. Another case of non-disjunction occurred by meiosis is Klinefelter syndrome. Klinefelter syndrome, in this case, female has 22 plus XX in gamete oocyte, and then male has 22 plus. Then once you fuse them, and then the zygote has 2n equal 44 plus XXY. Okay? We call this Klinefelter syndrome. It categorized into male, but this male has one extra X chromosome. Most of these males show no symptom, although some may show mixed secondary sexual characteristics, such as the breast developed, okay, and then the hip is broadening, and the testis, the size of the testis is small. And then they are sterile and may have a subnormal intelligence and may have a female body characteristics such as enlarged breast. So like this, you can see some breast development like a woman and has one extra X chromosome. And then the testis is very small, abnormally small testis, and also they are sterile. And then the probability is 1 out of 500 male birth. So Klinefelter syndrome, if you look at sex chromosome, it is going to be one more extra chromosome. Okay? So 3 copies of a particular chromosome. And non-disjunction of autosome. So far, we study non-disjunction of sex chromosome, but non-disjunction of autosome can occur during meiosis in the father or mother. Okay? So autosome, let me remind you, the 2n equal 44 plus XX and 2n equal 44 plus XY, these 44 chromosomes we call autosome. Okay? Resulting in eggs or sperm that are missing an autosome or that have 2 copies of an autosome. Fusions of these gametes with a normal sperm or egg result in a zygote with 1 or 3 copies of an affected autosome. So, for example, is trisomy 21, in other words, Down syndrome. Down syndrome has 3 copies of chromosome 21. When I say chromosome 21 means the homologous pair, we group them and put the numbering, and then 21st of homologous pair. We call that chromosome 21. So chromosome number 23, then it will make 46 chromosome, right? But so each chromosome number has 2 chromosomes as a homologous pair. But on chromosome 21, there are 3 chromosomes, not 2 chromosomes. Okay, we call that trisomy, 3 chromosomes at chromosome 21. In other words, Down syndrome. And Down syndrome includes this kind of characteristic, very weak muscle tone and a small mouth, and then the mouth is usually open because, to accommodate the tongue, and then the eyelid has a little bit distinctively shaped. Okay, so also they have a little bit heart formation is not normal, and they can be easily infected. So Down syndrome, you can write down this way. Down syndrome can occur to female and male, both gender. But only this part, the number of autosomes is different. Sex chromosome, there's no problem on the patient with Down syndrome. Autosomal chromosome number, abnormal. Correlation between maternal age and incidence of Down syndrome. In other words, when mom has baby at late age, maybe late, early in 50s or late in 40s, and then have a mom become pregnant, and then there's some high probability to have a baby who has Down syndrome. And then the skeletal system is weak, so they are short and have a poor muscle tone. So like this way. So you can see the small mouth held open partially because they want to accommodate the tongue. And then if you look at eyelid, this eyelid part, and then it's a little bit distinctly shaped. Okay, so there's some question about do Down syndromes have a shorter lifespan? And then until early 1980s, the average lifespan of a person with Down syndrome is about 25 years. It's pretty short. But scientists and medical doctors set up some institution and then provide well care to people with Down syndrome. So today, the average lifespan of a person with Down syndrome is approximately 60 years. It's still shorter than normal people, but longer than 25 years. Look at this karyotype. Can you see chromosome number 21? Okay. Each chromosome number has 2 chromosomes, such as homologous pair. But chromosome 21, it has 3 chromosomes, not 2 chromosomes. This one extra copy of chromosome on chromosome 21 makes people with Down syndrome. Okay, so the more chromosome number does not mean good. Just exactly 46 chromosome, that makes people normal. So this is a non-disjunction and aneuploid. That means the non-disjunction can occur during meiosis, even sometimes mitosis too. And then when the daughter cell has abnormal chromosome number, we call that aneuploid. Okay, let's solve the questions. Question 1. A typical human body cell contains how many sex chromosomes? This question is asking about the number of sex chromosomes in the cell, not the entire number of chromosomes inside the cell. So you can find the answer easily. Second question. What is it called the fusion of a gamete to form a new cell? So this question is asking about biological term. So the fusion of a gamete, such as egg and sperm, fuse together to form a new cell called zygote. What is it called, this phenomena? So you can find the answer from the lecture. This is the example of submitting week 4 assignment. So this is my email address, and include your email address, subject, Science 101 week 4 assignment. In main text body, you write down your last name, comma, followed by first name. And in main text body of email, you write down answer, question 1, question 2. The deadline of submitting this assignment is next Wednesday, midnight. So this is the end of lecture meiosis. Thank you.